Adipose tissue has emerged as one of the body's most immunologically active organs, fundamentally reshaping how we understand the relationship between fat, inflammation, and health. While commonly framed in terms of metabolic storage, adipose tissue is also a complex immunological environment, populated by diverse immune cells that respond dynamically to physiological stress, infection, and metabolic fluctuations. Far from being a passive bystander, adipose tissue plays an active and sometimes protective role in immune regulation. It produces a wide spectrum of cytokines and chemokines, both pro- and anti-inflammatory, and provides a site for immune cell activation, surveillance, and coordination. Rather than being inherently harmful, the immunological functions of adipose tissue are adaptive and essential to maintaining both metabolic and systemic balance. At the cellular level, fat tissue contains a rich network of immune cells, including macrophages, dendritic cells, natural killer cells, neutrophils, eosinophils, mast cells, and multiple T and B cell subsets. These immune cells are not random invaders but resident participants, deeply integrated into adipose biology. They respond to nutritional states, endocrine cues, and even microbial signals, adjusting their behavior accordingly. In lean, healthy adipose tissue, immune cells tend to exhibit an anti-inflammatory phenotype. For instance, M2, like macrophages, secrete IL-10, an anti-inflammatory cytokine that promotes insulin sensitivity and tissue repair. Similarly, regulatory T cells, TREGs, help maintain immune tolerance and metabolic homeostasis. This baseline immunological environment is not only compatible with metabolic health, it supports it. Adipocytes themselves are not merely structural or storage elements in this process. They actively communicate with immune cells through cytokine release and lipid mediators. Key adipokines like adiponectin and leptin exert immunomodulatory effects. Adiponectin, for example, inhibits the activation of pro-inflammatory NF-B signaling pathways and reduces the expression of TNF-A and IL-6 in macrophages. It also enhances insulin sensitivity and promotes a more quiescent, anti-inflammatory immune tone. Leptin, meanwhile, plays a more activating role, promoting T-cell proliferation and cytokine secretion in response to infection or energy deprivation. These dual roles reflect a nuanced regulatory architecture. Adipose tissue can scale up immune vigilance when needed, such as during infection or stress, but can also promote resolution and balance when the threat has passed. The pro-inflammatory activities often associated with adipose tissue, particularly in obesity, are not inherent to fat cells but are responses to physiological stress, usually chronic energy overload. In this state, adipocytes enlarge, hypertrophy, become hypoxic, and sometimes undergo necrosis. These stress signals trigger an adaptive immune response, leading to the recruitment of M1, like macrophages and other pro-inflammatory cells. This response is an attempt to repair or contain the damage, not a default state of fat tissue. While prolonged, unresolved inflammation can contribute to insulin resistance and tissue dysfunction, the inflammatory machinery itself is an evolved defense mechanism. It is the chronicity of the stimulus, not the presence of fat, that turns this response maladaptive. Recent research shows that this inflammatory process is reversible. Caloric moderation, exercise, cold exposure, and certain dietary interventions can restore a more anti-inflammatory immune profile within adipose tissue. Exercise, for example, not only reduces fat mass, but also increases the abundance of anti-inflammatory macrophages and TREGs in adipose depots. Nutrients like omega-3 fatty acids and polyphenol have also been shown to dampen pro-inflammatory signaling and support immune tolerance. These interventions improve both local adipose health and systemic metabolic function, reinforcing the idea that immune modulation within fat is not only possible, but therapeutically promising. Furthermore, the immune activity of adipose tissue may confer benefits beyond metabolic regulation. As an immune organ, adipose can mount responses to systemic infections and contribute to the clearance of pathogens. Fat-resident B cells, for instance, can produce antibodies, while macrophages and dendritic cells present antigens and initiate adaptive immune responses. Some studies suggest that adipose depots serve as reservoirs for memory T cells, providing immunological surveillance against reinfection. This immune competency may be particularly important during aging, when other immune organs such as the thymus decline in function. Adipose tissue, which remains throughout life, could serve as a compensatory immune niche that helps maintain protective immunity in older individuals. Viewed through this lens, adipose tissue is not merely tolerable, it is indispensable. Its immunological functions are integrated with its metabolic, endocrine, and regenerative roles, forming a tightly coordinated system that balances nutrient status with immune readiness. Dysfunction arises when this balance is lost, not when fat exists. The goal, then, is not to eliminate adipose tissue or suppress its immune activity, but to support its natural capacity to shift between pro- and anti-inflammatory states as needed. Promoting adipose tissue health through lifestyle, nutritional, or pharmacological means may be one of the most powerful ways to harmonize immunity, metabolism, and overall systemic resilience. In sum, fat is not a source of inflammation by default, it is a regulator of it. When functioning optimally, adipose tissue acts as an immune sentinel and peacekeeper, able to defend, repair, and adapt to changing internal and external environments. Recognizing this function opens up a new, more hopeful view of adipocytes, not as saboteurs of health, but as dynamic partners in immune balance and long-term vitality.